Okay. Okay. So my name's Jeff Kaplan. Today I'm speaking on behalf of the Centralized Research Instrumentation Core, known as CRIC, which is part of the Delaware INBRE program. I'm also the director of bioimaging, which is one of the core facilities, excuse me, in the CRIC network. And I'm also an R1 funded associate professor. So I do run a research program as well. And so, as someone who runs a research program, it's really essential that you use the latest techniques and the latest equipment. And the CRIC is a big component of that. By utilizing the core facilities, you'll make your grant applications much more competitive. So the CRIC is actually part of a larger network of shared resources. One being the CRIC is focused really on instrumentation. And there's a new component of DVI called the KRISTIN, which is Centralized Research Support Network, and that's directed by Claudine Jerkovich. And so where the CRIC is focused more on instrumentation, the KRISTIN is actually focused more on expertise and access to data. So let me just briefly mention the KRISTIN here. You can go to the INBRE website and get a little more information on it. But the KRISTIN is a newly formed network to provide access to biostatistics, epidemiology, study design, editorial service, and recently we added access to Medicaid data through the Center of Community Research and Services. So we expect this network will increase in size significantly over INBRE 4, this new phase of INBRE. Okay, so the KRISTIN, here's an overview of all the core facilities within CRIC. So there's quite a bit here. Things that are related to imaging, such as my bioimaging core, have an orange dot next to them. So up top here, let me open up a pen here. Up top here, there's other imaging cores, such as the Delaware State University Imaging Facility run by Hassan Bukhari. There's different types of whole organism imaging, including things for human imaging, such as translational PET imaging, as well as small animal PET. And in the Center for Biomedical and Brain Imaging, there's functional and structural MRI, as well as a new 9.4 Tesla small animal MRI system. Another big component is bioinformatics. We have the Center for Bioinformatics and Computational Biology, run by Sean Paulson. And we also have the bioinformatics core at Nemours. There's a DNA sequencing and genotyping center at DVI as well, run by Bruce Kingham. And somewhat related is the Gene Editing Institute at Christiana, as well as the Biomolecular Core at Nemours, which also has singular sequencing and still has microarray technology. So that's a general overview of these three main components, but there are other core facilities that have different technologies in them, such as histochemistry and tissue processing, which is basically a histology core that kind of feeds into some of these other imaging cores. The cell science core that has everything from flow cytometry to mass spectrometry, as well as a high throughput screening and drug discovery lab. So there are two, you can see it's kind of divided into two types of cores. There's the DE&BRE partner cores, which are up top here, and then there are the affiliated cores. So the DE&BRE partner cores are part of DBI, the Delaware Biotechnology Institute. And you could get the contact information here, as well as the websites, which will give you more information, and the directors that can help you with your projects. Okay, so I'm going to highlight my core just a little bit. Our core facility has 6, actually now 7 full-time staff members as of a couple weeks ago. They have expertise in all different technologies, everything from serial block phase scanning electron microscopy, Gene Ross here, standard scanning electron microscopy, laser capture microdissection, confocal microscopy, and single molecule image analysis, atomic force, Shannon-Chalban-Igam, and transmission electron microscopy and advanced sample preparation, Shannon-Matla. So it's quite a wide variety of services. So how do you get access to these services? Well, I'm going to explain how you get access to the Bioimaging Center, which would be quite similar to most of the other core facility as well. First, you would contact me to discuss the services you need. You can do that either by giving me a call or shooting me an email. And then determine if the services require routine or exploratory. So for routine services, I'll probably place you in contact with the correct bioimaging staff expert. For exploratory studies, or if you're not quite sure what you need, often we require a meeting. So it can be a consultation meeting with multiple staff members. If you really don't know what you want to do, then I'll invite you into one of our lab meetings. And so you'll give a presentation on your lab, the types of projects you have in your lab, and we'll discuss different ways that imaging can help your projects. So once we get to that stage, you can register your lab or yourself into a lab with our core facility management software, ILab. And if you have any questions with ILab, you can contact Debbie Powell in the Bioimaging Center. She can help you, or Adele Paoli at DVI. She can help you with the other partner cores. So at that point, we would just begin your project and either submit some samples or schedule time with Bioimaging staff. So ILab, you can access ILab here, udel.ilabsolutions.com. And this is actually a portal for a bunch of different core facilities. If you're at UD, you can use your UD credentials, and all other partner institutions can sign in here. So within ILab, you'll see a good percentage of those core facilities that I mentioned a couple slides ago are actually within ILab. Here's just a blow-up showing you the ones at the University of Delaware. There are some core facilities that aren't currently in the INBRE network that we'll probably try to get into the INBRE network, such as the NMR core and mass spec core over in chemistry. So I also want to mention that, just end this presentation with that DVI is moving. So DVI is currently here in the Delaware Technology Park. It will be moving to the Star Campus here in the new AP Bio building. Just a blow up of that map right here, and this is what the building will look like. And now I'm showing you a diagram. I really could probably just show you a picture now. That building is fairly far along now, and we expect that we'll be moving into this building starting in January, spanning all the way until April. And so the core facilities will probably move on the latter half of that. We'll move some essential things with various faculty members that need them, that are going in early, and then the majority of it will probably move closer to March and April. Here's a floor plan. In red here is the Bioimaging Center. There's the CBCB is right next door here. This is a outreach lab. The Genomics Center is here, and the Bio-IT, which houses the Biomix cluster, also partially supported by INBRE, is housed here. This cluster actually provides support for all three of those partner cores that I just described. Okay. So expect that there will be a little bit of downtime during that move. We're hoping to make it as fast and smooth as possible, but we do have quite a bit of complicated instrumentation, both in the imaging core and the sequencing core. And it's going to take a while also to move all those servers, minimizing that downtime. So the last thing I want to mention is that the Bioimaging Center is opening up 2 new satellite core facilities that are located on main campus. And they're located in the same building as the MRIs, the Center for Biomedical and Brain Imaging, the CBBI. In that space, there will be a new fax flow facility. We just hired a new associate scientist, Rich West. As I mentioned, we now have 7 people. Rich joined us a couple weeks ago, and he'll be leading the flow cytometry initiative, not only at this facility here, but also at various places across campus, including DVI. There's also a multimodal animal imaging facility there. We have 2 instruments that were recently put in that space. One is an upright intravital multiphoton microscope, and the other one is a micro CT. And we already have very good access to the intravital system. We're buying the anesthesia equipment for that now, and it should be delivered very soon, within a couple weeks, I would say. And the micro CT will probably be integrated into the Bioimaging Center completely by the end of July. If you need access to that in the meantime, just feel free to give me a call, and I'll help you get access to it. Okay, and the last thing I want to mention, thank you, Dawn, for mentioning this in the beginning, is the Core Center Access Award Program. And we'll be starting this up very, very soon. This Core Center Access Award Program provides up to $4,000 for 6 months of access to one of those core facilities within the network, as well as there will be new Core Center Access awards that are specifically geared toward the KRISTIN, and we'll have more details on that soon. I really encourage people that are trying to collect preliminary data for grants, specifically NIH grants, to take advantage of this program, as well as people that are in between funding cycles or need that last piece of data on a project that was just finished and you need to get that publication out. This is a really great program where it brings funding for both new investigators as well as established investigators access to this network of core facilities. And so with that, I'll take any questions.